Welcome students to an inspiring venture into subcellular fractionation, where you will unravel the secrets of cellular components through differential and density gradient centrifugation. Prepare to explore these transformative techniques, connecting foundational cell biology with cutting-edge applications in research and medicine. Learning objectives. By the end of this course, you will be able to explain the basic principles and importance of subcellular fractionation, master techniques for subcellular fractionation, predict organelle separation, outcomes under different centrifugation conditions, compare and contrast differential and density gradient centrifugation, recognize challenges and modern advances in organelle isolation techniques. Apply practical skills to overcome fractionation challenges. Identify key research and medical applications of subcellular fractionation. Cells, the fundamental units of life, are highly organized structures containing specialized organelles like the nucleus, mitochondria, ribosome, endoplasmic reticulum, Golgi bodies, lysosomes, and chloroplast in plants, performing critical functions like energy production, protein synthesis, and intracellular digestion. Understanding cellular function, scientists study these components individually through subcellular fractionation, a technique that separates organelles for detailed biochemical and structural analysis. Central to this process is centrifugation, which employs centrifugal force to separate cellular parts based on size, mass, or density. Two classic methods dominate: differential centrifugation, which sequentially sediments component, and density gradient centrifugation, which achieves higher resolution based on bion density. Pioneered by Claude, Palade, and Dove, whose Nobel Prize-winning discovery of lysosomes revolutionized cell biology. Mastering these principles bridges basic cell structure with modern molecular biology, making subcellular fractionation a cornerstone in proteomic research, drug discovery, and biomedical innovation. Subcellular fractionation. Subcellular fractionation is a pivotal technique in cell biology, enabling the isolation of cellular components such as organelles, membranes, and macromolecules for detailed study. Fractionation refers to breaking a complex mixture such as homogenized cell suspension into its individual parts or fractions. Each fraction is enriched in a particular organelle or molecule. By analyzing these fractions separately, scientists can pinpoint the location of enzymes, proteins, lipids, or nucleic acids in the cell. Preparation for subcellular fractionation. Before centrifugation can be performed, cells must be disrupted carefully to release their contents without damaging organelles. Cell disruption or the homogenization methods include the mechanical methods, grinding with mortar and pestle, homogenizers, blenders, French press. Sonication, high frequency sound waves break open the cells. Osmotic shock, placing cells in hypotonic solutions, causing them to burst. Enzymatic digestion, lysozyme for bacterial cell walls, cellulase for plant cell walls. Homogenate preparation, the broken cell suspension is called the homogenate. It contains all organelles, proteins, and cytosolic components in a mixed form. Buffers and stabilizers to maintain the organelle integrity, Buffers such as sucrose, mannitol, or phosphate solutions are used. These prevent osmotic damage and maintain pH. Protease inhibitors are often added to prevent degradation of proteins. Sedimentation coefficient, or the Swedberg unit, the rate of sedimentation is often expressed in Swedberg units, represented by S, where 1 Swedberg is 10 raised to power minus 13 seconds. Ribosomes, for example, are classified as 70S in prokaryotes or 80S in eukaryotes, indicating their sedimentation behavior. Importance of subcellular fractionation in research and analysis. To determine the location of biochemical pathways, for example, Krebs cycles in mitochondria, to study the organelle structure and function, to purify components for proteomics and genomics, to investigate disease mechanisms where defective organelles are implicated. Applications of subcellular fractionation. Subcellular fractionation has broad applications. Biochemistry, identifying enzyme location, for example, Krebs cycle enzymes in mitochondria. Fractionation aids in subcellular localization studies, determining where proteins or signaling molecules reside, which is vital for understanding pathways like apoptosis, for example, cytochrome C release from mitochondria. Molecular biology, isolating ribosomes for protein synthesis studies. Medical research, studying defective lysosomes in storage disorders. Genomics and proteomics, analyzing organelle-specific proteins, plant and animal breeding. Isolating chloroplasts, mitochondria for genetic engineering. Challenges of subcellular fractionation. Challenges include optimizing homogenization to avoid organelle damage and minimizing contamination or cold conditions required. Gentle lysis preserves fragile structures like Golgi, which buffers with sucrose or mannitol maintain osmolarity. Protease inhibitors prevent protein degradation, and cold conditions at 4 degrees Celsius reduce enzymatic activity. Verification is crucial as impure fractions can skew results. For example, contaminated mitochondrial fractions may include endoplasmic reticulum, affecting metabolic studies. Its integration with modern techniques like mass spectroscopy enhances its utility, making it indispensable for advancing cell biology and related fields. Maintaining organelle integrity, avoiding cross-contamination of fractions, correct interpretation of data, controls essential. High cost and need for technical expertise. Advances in fractionation. Ultracentrifuge speeds up to 5 lakh G. Immunoisolation using antibodies attached to beads to pull down specific organelles. Magnetic beads and nanoparticles for rapid purification. Automation. Robotic fraction collectors for high throughput research. Centrifugation. Centrifugation is driving force behind subcellular fractionation. The principle is based on Stokes' law, which describes how particles sediment in a fluid under gravity or centrifugal force. Centrifugation involves spinning a sample at very high speeds, generating a strong centrifugal field that drives particles outwards. The rate at which particles move depends on their size and shape. Larger and denser particles sediment faster. Bion density. Particles move until they reach a medium of equal density. Viscosity of medium. Higher viscosity slows sedimentation. Types of centrifuges include tabletop for low speed, high speed for microsomes, and ultracentrifuge for viruses or ribosomes reaching over 1 lakh G. Differential centrifugation. The principle of differential centrifugation is advanced technique in cell biology and biochemistry, widely used for separating cellular components based on their physical properties. This method is fundamental for undergraduate students to understand as it provides a practical approach to isolating organelles, membranes, and macromolecules, enabling detailed studies of cellular function. By exploiting differences in size, density, and shape, differential centrifugation separates cellular constituents through sequential spins at increasing centrifugal forces. Its simplicity and accessibility make it a stable in laboratories, particularly for initial fractionation before more refined techniques like density gradient centrifugation are applied. Step-by-step process of differential centrifugation. Homogenization. The process begins with homogenization, disrupting cells to release their contents into a suspension called the homogenate. Common methods include Potter-LWM homogenizer, a glass tube with a Teflon pestle, ideal for soft tissues like liver or brain. Downs homogenizer uses a glass pestle for gentle disruption, suitable for cultured cells. Sonication or blenders for tougher tissues. Though these risk organelle damage, homogenization is performed in an isotonic buffer, for example, 0.25 molar sucrose or mannitol with a pH of around 7.4 to maintain organelle integrity. Buffers often include protease inhibitors, for example, PMSF to prevent protein degradation and EDTA to chelate metal ions that activate nucleases. Sequential centrifugation steps. Differential centrifugation involves sequential spins at increasing speeds, with each step pelleting specific components while leaving others in a supernatant. A typical protocol is low speed centrifugation at 600 to 1000 G for 10 minutes. Pellets large, dense structures like nuclei and unbroken cells. For example, at 800 G, nuclei from liver tissue form a pellet, while mitochondria remain in the supernatant. Medium speed centrifugation, which is 10,000 to 20,000 G for 15 to 20 minutes. The supernatant is spun to pellet mitochondria, lysosomes, and peroxisomes. A 15,000 G spin isolates mitochondria from rat liver. Now, the high-speed centrifugation, which is around 50,000 to 1 lakh G for 30 to 60 minutes, pellets microsomes like endoplasmic reticulum and Golgi fragments and small vesicles. This requires an ultracentrifuge. Ultracentrifugation, which is greater than 1 lakh G for 1 to 2 hours, isolates ribosomes, viruses, or large protein complexes, leaving cytosolic proteins in the supernatant. Pellet washing and resuspension. After each spin, the pellet is washed by resuspending it in fresh buffer and recentrifuging to remove the contaminants. For example, a mitochondrial pellet may contain endoplasmic reticulum fragments, so washing at 15,000 G improves purity. Supernatants are carefully decanted or pipetted to avoid disturbing pellets. We will now discuss about advantages of differential centrifugation, simple and widely used. Quick isolation of basic organelle fraction. Simplicity requires basic equipments like the tabletop or high-speed centrifuges and straightforward protocols, accessible for teaching labs. Speed. Initial separations are quick with spins lasting for 10 to 60 minutes. Versatility applicable to various tissues like the liver or the brain, and cell types like yeast mammalian cultures. Cost effective, no need for expensive gradient media. Now, the limitations of differential centrifugation. Fractions are not pure. Organelles of similar size may contaminate each other. Cannot distinguish between similar density organelles, for example, lysosomes versus peroxisomes. Low resolution. Components with similar sedimentation rates, for example, mitochondria and lysosome, co-pellet, leading to contamination. For example, a 15,000 G spin may include peroxisomes with mitochondria. Limited purity requires subsequent techniques like density gradient centrifugation for refined fractions. Mechanical stress. High speeds can damage fragile organelles like Golgi. Operator skill. Inconsistent homogenization or improper speeds skew results. Contamination is a major challenge. For instance, microsomal fractions may include Golgi or plasma membrane vesicles. Over-homogenization can fragment organelles, while under-homogenization leaves cell intact, reducing yield. Students must optimize conditions through trial and error, guided by marker assays. Additionally, tissue-specific differences, for example, fibrous muscles versus soft liver, affect homogenization efficiency. Now, the applications of differential centrifugation. Differential centrifugation is used to isolate specific organelles for functional studies. Nuclei for DNA extraction, transcription factor analysis, for example, studying histone modifications in cancer research. Mitochondria for respiration studies, ATP synthesis or apoptosis. For example, cytochrome C release in cell death pathways. Lysosomes for enzyme activity assay. For example, acid hydrolases in lysosomal storage diseases. Microsomes for studying endoplasmic reticulum protein folding or drug metabolism by cytochrome P450 enzymes. Peroxisomes for fatty acid oxidation studies. We will now discuss about density gradient centrifugation principle. Density gradient centrifugation is a sophisticated technique in cell biology and biochemistry that separates cellular components with high precision based on their bond density. Unlike differential centrifugation, which relies on size and sedimentation rate, density gradient centrifugation uses a density gradient medium to allow particles to migrate to a position where their density matches that of the surrounding medium. Developed in the mid-20th century, density gradient centrifugation gained prominence through experiments like Michelson-Stahel's study of 1958, which confirmed semi-conservative DNA replication using cesium chloride gradients. Its ability to achieve high-resolution separations makes it indispensable in research from virology to proteomics. Preparation of gradient media for density gradient centrifugation. Gradients are prepared as continuous, smooth density transition or step discrete layers. Continuous gradients use a gradient maker mixing high and low density solutions. For example, 40% and 10% sucrose. Step gradients are layered manually, for example, 20%, 30% or 40% sucrose. Self-generating gradients form during centrifugation with percol. Sucrose gradients, most common for organelles. Sucrose is widely used due to its affordability and osmotic properties. Linear, for example 10 to 40%, or step gradient, for example 20% to 40%, are common for organelle separation. However, high concentrations increase viscosity, slowing the sedimentation. Cesium chloride gradients used for nucleic acids. Percol, alternative density media. Percol, a colloidal silica coated with polyvinyl pyrrole iodine, is inert and forms self-generating gradients during centrifugation. It's ideal for cell separation, for example, lymphocytes, due to low osmolarity and biocompatibility. Ficol. Ficol, a polysaccharide, is used for cell separation, particularly peripheral blood mononuclear cells. Sample loading for density gradient centrifugation. The sample, for example, cell homogenate, is layered atop the gradient or mixed within for isopycnic runs. Gentle pipetting prevents mixing. Centrifugation swinging bucket rotors are preferred for gradients, ensuring particles band at tube bottoms. Rate zonal runs are short 1 to 4 hours, 50,000 to 1 lakh G, while isopycnic runs are longer 12 to 48 hours, 1 lakh to 2 lakh G. Types of density gradient centrifugation. First is the rate zonal centrifugation. Rate zonal centrifugation separates particles based on size and shape during a short centrifugation run before they reach their Bion density. Particles move through the shallow gradient, for example 5 to 20% sucrose, at different rates based on their sedimentation coefficient, which is represented by S. For example, ribosomes, which are 70S, separate from polysomes in a sucrose gradient at 1 lakh G for 2 hours. This method is ideal for macromolecules or organelles with distinct sizes but similar densities. Next is the isopycnic centrifugation. Isopycnic centrifugation involves prolonged centrifugation until particles reach equilibrium at their Bion density. Dense media like cesium chloride or percol form steep gradients, for example 1 to 1.8 G per centimeter cube. DNA with a specific Bion density bands in cesium chloride at 1,50,000 G after 24 to 48 hours. This method excels at separating particles with similar sizes but different densities, such as mitochondrial subtypes. Used to demonstrate semi-conservative replication, which is the Michelson-Stahls experiment of 1958. Fraction collection. Post-centrifugation, fractions are collected by puncturing the tubes or using automating fractionators. Bands are visible in dense media like cesium chloride or stained for visualization. Next are the organelle purification. Isopycnic centrifugation purifies mitochondria, lysosomes or peroxisomes. For example, Percol gradients separate mitochondria from endoplasmic reticulum contaminants in liver homogenates. Virus purification. Cesium chloride gradients purify viruses like adenovirus at around 1.34 g per centimeter cube for vaccine production or virology studies. Cell separation. Phi-Colp gradients isolate PBMCs for immunology research, separating lymphocytes from denser erythrocytes. Macromolecule separation. Rate zonal centrifugation separates RNA, ribosomes or protein complexes in sucrose gradients for structural studies. Analysis. Fractions are analyzed via microscopy, enzyme assay, for example, acid phosphatase for lysosome or Western blotting to confirm purity. Applications of density gradient centrifugation. Isolation of mitochondria, lysosome, peroxisomes with high purity. Separation of RNA and DNA. Purification of viruses. Studying membrane proteins. Now, we will be discussing about the advantages of density gradient centrifugation. High purity. It separates components with close sedimentation properties, for example, lysosomes versus mitochondria. Versatility. It is applicable to cells, organelles, viruses and macromolecules. High resolution. Continuous gradients distinguish subtle density differences. Biocompatibility. Media like Percol are gentle for live cells. High resolution and purity can distinguish organelles of similar size but different densities. Limitations of density gradient centrifugation. First is that it is more time consuming than differential centrifugation. Next is it requires expensive ultracentrifuges and expertise. Also, it is time intensive. Isopicnic runs can take about 24 to 48 hours. Equipment needs. It requires ultracentrifuges and gradient makers. Next is the complexity. Gradient preparation and fraction collection demand skill. Next is the cost. Media like Percol or cesium chloride are expensive. Dear students, we will now discuss about the comparison between differential and density gradient centrifugation. The first comparison is on the basis of separation. The differential centrifugation, size and sedimentation rate in a homogeneous medium. But in density gradient, bond density in a gradient medium. Next is the resolution. For differential centrifugation, it is low due to co-sedimentation of similar particles. For density gradient centrifugation, it is high and separates particles with close densities. Next is the time requirement. For differential centrifugation, it is short, 10 to 16 minutes per spin. But for density gradient, it is longer, around 1 to 48 hours. Next are the application. Differential centrifugation is used for initial isolation, for example, nucleus, mitochondria. Density gradient centrifugation is used for purification, for example, viruses, PBMCs. Equipments. In differential centrifugation, tabletop or high-speed centrifuges are used. But in density gradient centrifugation, ultracentrifuges or gradient markers are used. Next is the complexity. In differential centrifugation, it is simple and minimum steps are required. But in density gradient centrifugation, it is complex and requires gradient preparation. Next is the cost. Differential centrifugation is low cost and uses standard buffers, but the density gradient is high cost and uses specialized media, for example percol or cesium chloride. Next is the gradient use. In differential centrifugation, no gradient is involved and is a uniform medium, but in density gradient centrifugation, it uses continuous or step gradients. Next is the purity. Differential centrifugation is prone to contamination, for example, endoplasmic reticulum with mitochondria. But the density gradient centrifugation, it achieves higher purity with distinct banding. Last are the examples. The differential centrifugation involves isolating mitochondria from liver at 15,000 G, but in density gradient, purifying DNA in cesium chloride gradient, which is the Michelson-Stahls experiment. In conclusion, subcellular fractionation is a cornerstone of modern biology, allowing the separation and detailed study of organelles, macromolecules, and genetic material. Differential centrifugation offers quick and simple isolation of organelle-enriched fractions, while density gradient centrifugation provides high-resolution purification based on bion density. Together, these techniques have revealed fundamental truths about cell biology, from the discovery of gene mapping to understanding diseases and developing new therapies. Dear students, as you master the subcellular fractionation, remember that understanding this process is not just about learning a laboratory technique, but about uncovering how life organizes itself at the microscopic level. Mastery of these principles will strengthen your foundation in cell biology and open pathways for deeper studies in molecular research and medical science. Thank you for listening patiently.